so now we're going to go through paper 6, which is the alternative to the practical for biology. And this is IGCSE from Cambridge International Examination, CIE, from May-June, summer 2018. And the code is 0610/62. It's variant 2. All right, let's begin.
Darn. Oh.
Question 1. A student investigated the rate of respiration of yeast at 2 different temperatures. Step 1. The student was provided with a yeast suspension which also contained glucose. 2 identical sets of equipment were set up as shown in figure 1.1. This is figure 1.1. Okay, we have a test tube, which is just water, and that water will be kept at a particular temperature. Inside this pipette, we have the yeast with glucose, so it can respire. And as it respires, it produces bubbles. And they just count the bubbles. Step 2. One set of the equipment shown in figure 1.1 was placed into a warm water bath, and the starting temperature of the water bath was recorded as 50 degrees Celsius, and the other set was placed into a cool water bath, which had a starting temperature of 25 degrees Celsius. The volume of water in each water bath was the same. The student counted the number of bubbles, gas bubbles produced by the yeast suspension in 5 minutes and recorded the results in a tally chart. This is shown in figure 1.2. Okay, this is your tally. Keep in mind that when you record data, this is your rough work, but they never want tallies in the final work. Okay, so your table has to have actual numbers. A1. Prepare a table and record the student's results shown in figure 1.2. Well, this was quite a straightforward table. So one of the headings is just the temperature of the water bath in degrees Celsius. And the other heading is the number of bubbles. So the number of bubbles in 5 minutes. Okay, and we had 2 different temperatures. We had one at 25 degrees Celsius and one at 50 degrees Celsius. So now that I have this information, I will put the lines in, and then I will know that when I put the results, they will line up with the temperature that they're supposed to. Okay, and while the lines don't have to be absolutely perfect, they do have to be drawn with a ruler, and you have to draw in all the lines, including the ones along the outside. So if we look at our tally, the cool water had 7 tally marks for 7 bubbles, and the warm water had 5, 10, 15, 19 tally marks for the 19 bubbles. So that's 7 bubbles and 19. And do not write in any units in the table or you lose a mark. All right, that's it. That's all they want. Step 4. At the end of the investigation, the final temperature of the water in both water baths was measured. The results are shown in figure 1.3. Okay, so they're certainly not 50 degrees and 25 degrees. They've definitely gone down. So this is figure 1.3. 2. Use the information in step 2 and figure 1.3 to complete table 1.1. So step 2 just said it started out at 50 and it started out at 25. Okay, so the starting temperature for the warm water bath was 50 degrees Celsius. Again, no units because it's in the heading. And the cool water bath was 25 degrees Celsius. The final temperature for the warm one, it went, these go up by 1 because there's 10 gradations between 40 and 50. So that's 41, 42, 43. That's 43 degrees Celsius. And here we have 21 degrees Celsius. And that's it. That's all they want. B1. State a conclusion for the results. Okay, so what we can see is that as the temperature has gone up, so did the number of bubbles produced, and that's because of respiration. So respiration increased as the temperature went up. So the warmer the water, the more bubbles are produced, so the more respiration has occurred. One thing you have to be aware of, though, is you can't just talk about one of the situation. If you'd said the yeast in the warm water bath have lots of bubbles, you also have to say the yeast in the cold water bath have few bubbles. Okay, but here you're saying basically as it gets warmer, there are more bubbles. So you're kind of talking about both. 2. Counting the number of gas bubbles produced may not be an accurate method of measurement. Explain why and suggest an improvement. So that improvement has to match the explanation. Okay, so the explain why, the reason why, whenever you're measuring bubbles, is that some of the bubbles may be little bubbles, some of the bubbles may be big bubbles. They still count as a bubble, but the amount of oxygen produced can be very different. So the bubbles may be different sizes, so they could be different volumes. Okay, and how do you improve on this? Well, counting the bubbles doesn't work as well. You need to actually measure the actual volume of bubbles produced. And a good way to do that is using a gas syringe. So use a gas syringe to measure the volume of bubbles produced. And while it might be obvious that the purpose of a gas syringe is to measure the volume of bubbles, just stating using a gas syringe isn't enough. You have to actually say what you're using it for, measuring the volume of the bubbles. Okay. 3. Identify a variable that should have been kept constant during this investigation, but was not, and suggest how this variable could have been kept constant. Okay, pretty much the main variable that should have been kept constant and wasn't is the fact that the water baths were not constant. They were obviously just left to cool down over time. So the water baths should have been kept at a constant temperature. And how this variable could have been kept constant? Well, the key word that they really like to see here is a thermostatically controlled water bath. That's the best way to do it. That's the best way to control the temperature. So the temperatures can be kept constant by using a thermostatically controlled water bath to keep the temperatures constant. And a thermostatically controlled water bath is just basically a water bath that's controlled by an electrical switch, basically. It will turn off if it's too hot. It will turn back on when it's too cold. And that's a much more reliable way of controlling the temperature. 4. Identify the variable that was changed, the independent variable, and the variable that was measured, the dependent variable in this investigation. Okay, so they changed the temperature from between 25 degrees Celsius and 50 degrees Celsius, and they measured the number of bubbles produced. They counted how many bubbles there were. So the temperature was what was changed, and the number of bubbles produced was what was measured. C. A scientist investigated the effect of different concentrations of glucose on the rate of carbon dioxide production in yeast cells. They measured the volume of carbon dioxide production by the yeast cells in 5 minutes. 1. The scientists decided to test their method before beginning the investigation. They performed 3 trials using one unknown concentration of glucose. The results are given in table 1.2. Okay, so they tried it 3 times in 5 minutes. They're fairly similar results, so they continued on with the rest of the experiment. Calculate the average volume of carbon dioxide produced in 5 minutes. Okay, so the average volume equals 13.6 centimeters cubed plus 14.3 centimeters cubed plus 12.9 centimeters cubed divided by 3. And the answer you get there is 13.6 centimeters cubed. Okay, units are already in, so you just write 13.6. And then we have to calculate the average rate of carbon dioxide production per minute. All right, so the average rate equals the average volume, that's 13.6 centimeters cubed, divided by the time. And it says it's in 5 minutes, so 5 minutes. And here it says centimeters cubed per minute, not per second, so you don't need to even convert it into seconds. Okay, when you type that into your calculator, you get 2.72 centimeters cubed per minute. Okay, and you can either leave it as 2.72 or you can just put it as 2.7. 2 or 3 significant figures is just fine. Okay. 2. The scientists performed the investigation. Their results are shown in table 1.3. All right, they have some data. And then you need to plot a graph on the grid using the data in table 1.3 to show the effect of glucose concentration on the rate of carbon dioxide production and include a line of best fit. So if we look at this, here we have to go from well, probably 0 to 3. And here we have to go from probably 0 to 5. All right, so let's look at our graph. Okay, so the independent variable is on the x variable, that's the thing you change, and you are changing the concentration of glucose, and the units are percent. So concentration of glucose in percent. And on the y-axis, you're measuring the average rate of carbon dioxide production in centimeters cubed per minute. Okay, so the average rate of carbon dioxide production in centimeters cubed per minute. And as we said earlier, for the concentration of glucose, we need to go from 0 to 3. And so the nice logical scale for this, we have 1, 2, 3, 4, 5, 6 large squares. So the logical number to go up by is 0.5s, 1.0, 1.5, 2.0, 2.5, and 3.0. Now, don't forget to use the same number of decimal places, really. So if you have 1.5, you should do it to 2.0. Okay, and on the y-axis, we're going from, we said from 0 to 5. And if we look at this, we have 1, 2, 3, 4, 5 large squares. That's nice and easy. 1, 2, 3, 4, 5. Can't get much more simple than that, really. Okay, now we just plot the data. Okay, so here are the points. If you notice, we have little x's, and they accept x's, they accept dots with circles, not like that, dots with circles around them. They do not accept large blobs. Yeah, if you drew a dot like this, you would lose a mark. That's not a good point. If you did a little tiny dot like this, little dot like that, you would lose a point because you can't really see it. Okay, and as soon as you draw a line through it, it's gone. Nobody knows if you drew the correct points. Okay, so a cross or a dot with a circle around it, those are good ways of drawing your dots. Those are the ways they accept. Now, your line, for this one, I would draw a smooth curve. You can actually go from 0, 0 for this one, but don't just, well, that's not good. But quite often you don't go all the way through 0, 0. Okay, so this line, I think that's actually pretty good compared to how I normally do them. First try. All right, so whenever you do these lines of best fit, you need to use a sharp pencil. Okay, and your dots as well. Make sure you use a sharp pencil. And don't draw really hard, because if you erase it, you need to make it clear that it's the new line. You don't want to have any sort of remnant of an erased mark behind. And the same goes with your drawings. All right, so this is a nice line of best fit. You could also actually have done a dot to dot. I try to stay away from dot to dots quite often, but this time they accept it because it's very clearly dot to dot. Although I think around here it wouldn't work as well. A straight line, it should be a curve. So they'll accept dot to dot, but you could also do smooth curve. I would do a smooth curve. One more thing to notice about this graph, when it levels off the top here, that does not mean the reaction has stopped. It means that the rate of reaction has stopped increasing. This is going for the rate of reaction, not how much has been produced. Okay, that's important for the next bit. 3. Describe the effects of glucose concentration on the rate of carbon dioxide production by respiring yeast cells shown in your graph. Okay, so what you can see is that as the concentration of glucose increases, the rate of the concentration of glucose increases. And it starts to slow down at about 2% of glucose, and the rate stops increasing at 2.5%. All right. So as the concentration of glucose increases, the rate of carbon dioxide production also increases. So the rate decreases at 1.5% glucose and levels off at 2.5% glucose. Okay, make sure your decimal places are very obviously noticeable when you're doing these, and you always have to put the units in. And this is the rate decreases, not the reaction decreases, because the reaction doesn't stop. Okay, and the rate increases from 13 centimeters cubed per minute to 4.4 centimeters cubed per minute. This last point is just a quoting data point. Okay, so this rate decreases at 1.5% glucose and levels off at 2.5% glucose. That is a very obvious trend in the graph, which you should include. But when it says describe the effect, quite often you get a mark just for quoting some specific data point. Okay, so let's say it says it increases from this value to this value. Again, make sure your decimal points are nice and clear. And include your units in. Don't just say from 1.3 to 4.4. All right, let's move on. 4. Estimate the concentration of glucose the scientists used in their test method using your answer for 1C1 and your graph to find this value. All right, so let's look at 1C1. So 1C1, you calculated the average volume produced in 5 minutes, and then you calculated the average rate of carbon dioxide production per minute. So we're going to use the second one. The average rate was 2.72 centimeters cubed per minute. So from 1C1, the rate equals 2.72 centimeters cubed per minute. All right, now we look at from the graph. From the graph, what is the percent at 2.72 centimeters cubed per minute from the graph? So at 2.72, or 2.7, because you can't measure that accurately on this graph, 2.7 centimeters cubed per minute crosses about right there, approximately there. Okay, and then if we go down from that value, we have this, it stops at this point. Okay, and remember each one of these small squares was 0.05%. So this value is 1.05%. And they know it's from the graph because you've drawn the lines. Don't just read it and say it's from the graph. You actually have to draw the lines so that the examiner knows that you know what you're actually looking at. So it's 1.05%. All right, and percent's already put in there. So make sure you draw your lines on the graph so that the examiner knows how you got your results. D1. Describe how the student could show that the glucose used in the investigation is a simple reducing sugar. Okay, the test for that is Benedict's reagent. And how do you go about doing the Benedict's test? Well, you heat it to at least 80 degrees Celsius and you check to see if there's a color change. So you heat it to 80 degrees Celsius and then you look for a color change. You look for a color change from blue to either green, orange, yellow, or red. And this shows the presence of reducing sugars. And the color it changes shows how much there is. 2. Identify one hazard when testing for simple reducing sugars and describe one precaution that could be taken to reduce the risk. Okay, so the precaution must actually match the hazard. And one of the big hazards is the fact you're using Benedict's reagent. So Benedict's reagent is an irritant. It's not going to be really terrible, but you don't want it in your eyes, and it's not really good to have on your skin. Okay, so the way you can protect yourself against that, you can reduce the risk, is to wear goggles, or you can say to wear gloves. So you can wear goggles or gloves. So another thing you could do is you could say you're using hot water. It's not boiling water, but it's still hot water. So you can use a thermostatically controlled water bath to make sure it stays as just hot water and doesn't boil. Doesn't go above 80 degrees. Okay, but you only write down one. You don't write down both of them. You only get marks for one anyway. And if one of them isn't perfectly correct, you lose the marks anyway. Question 2. Figure 2.1 shows Asteronella, which are microscopic algae that live in fresh water. Okay, so this is Asteronella. What you might not be able to tell is that each one of these arms is one algae, and they're all joined together in the center. All right. A1. Make a large outline drawing of the algae and do not label your drawing. This is my least favorite part of a biology exam because I find drawing to be very challenging, but you can do it with practice. If you're like me and find drawing challenging, what you need to do is just practice. Artists, though, often find this very difficult to do the drawing section as well, because artists tend to put in more detail than what is allowed. You're actually only allowed to do very minimal details, because this says a large outline drawing. Basically, you only draw the outline. You draw the outline. Basically, what you're doing is this. You're drawing each, well, not covering it up. You're doing a better job than this, and you're not drawing it on this drawing. But you just draw the outline. Okay? If you look at this, you can actually sort of see that there's individual cells in here. This is one individual cell, I think. I think this is another one. It's hard to tell. But you don't draw that. Okay? All you do is the outline of it and the circle in the middle. Other points of this, it says large. Now large means that it needs to take up most of the space you're given. It cannot go over any of the writing though. You can't make it too big. If it goes over the writing, that's too large and you lose marks. And it says do not label your drawing, which makes that a little bit easier as well. Okay, because you might not know any of the structures. That's okay. All right. Some very important points when you're drawing in biology. You use a sharp pencil for one, and you don't draw really hard, because when you make a mistake, well, not everybody does, somebody like me does, when you make a mistake drawing, you need to be able to erase it without having the line still showing through, because that causes confusion and the examiners don't like it. Other things, when you draw a structure, you need to make sure the ends touch where you shouldn't really be able to tell where you started and ended. If you draw a circle like that, you lose marks. If you draw a circle like that, it's hard to say if they take a mark off or not. Sometimes they might not. It's hard to say. So you draw very carefully so you can't lose marks. You don't shade in. You don't color in any sections. You don't do stipples or dots to show texture. You don't draw any internal structures if it says to do an outline. You don't feather. This is feathering. Feathering makes drawing nice and much easier. If you're drawing a leaf, a feathered leaf is probably a lot easier to draw than trying to draw one smooth line that looks exactly the same. This, you'll lose, you won't get any marks for the point for making a smooth outline. Okay, I think that is about the key bits. Oh, another thing with this diagram, with this one, it looks like these lines are very, very straight. Do not use a ruler. The examiner will be able to tell that you've used a ruler, and in real life, things aren't perfectly straight. If you look at this, you can sort of see that there's a little bit of bumps, which means not much. Well, that would be terrible, but a little bit of bumps, which means if you draw it with a ruler, it will look wrong. Okay? So do not use a ruler. Nature rarely lets you use rulers, although it is very straight. You just have to be careful trying to draw a very straight line, which is not very easy. Okay? You need to try and draw in the same proportions. So you have to draw 1, 2, 3, 4, 5, 6, 7, 8 points, and they have to be in the same, approximately the same direction, same spacing, same thickness, approximately. Okay? But the examiner knows you're a biology student, you're not an art student, so if it's not exactly perfect, they're not going to take marks off as long as you've done a pretty good job and have obviously taken care with your drawing. Okay, now I'm going to draw the drawing and you're going to see the final result rather than all my attempts. Okay, there we go. On first glance, this diagram looks like it should be a very easy one to draw. I found this one actually very tricky. So don't worry if you find it tricky. Just don't use a ruler, don't cover up the text, and try and draw it as straight as you can. It's very tricky to draw straight lines using this method that I'm using. However, doing it freehand is difficult to draw straight lines anyway. So if you notice that these lines, they line up. They should line up. They're meant to line up. So I found it easier to draw a circle in the middle and then draw a sketch of where the lines should be. But don't worry if you struggle with it. You just have to practice. Okay, so the marks you get for this, one of the marks is that you have one single clear outline with no shading. One is that you've done an appropriate size, at least half of the space that's available. And you have to make sure that it doesn't actually go over any writing. You want to make sure that there are 8 arms and that there is a center circle drawn. And that's what you get the marks for. Okay, and the 8 arms are in approximately the right position. So if it's not 100% perfect, it's not worth losing other marks on your exam because you'd have to spend half an hour on this one picture. Okay, 2. Measure the length of the line AB in figure 2.1 and include the unit. Okay, the length of AB is this line here. You just use your ruler, measure from the ends, and include the unit. So when I measured this line, I got 85 millimeters. But you could have got 84 or 86, or you might have got a slightly different number because you printed off a PDF onto a different size piece of paper. But as long as you know how to do the measurement, you should be okay. On the actual day, everybody will have the same piece of paper printed off to the same scale, so you'll be fine. If you'd written down 8.5 centimeters, that's okay. You just have to include the units. So that was 85 millimeters. And use the formula to calculate the actual diameter of the algae shown in figure 2.1 and include the units. So magnification equals length of the line AB divided by the actual diameter of the algae. We're looking for the actual diameter of the algae, which would mean that it equals the length of the line AB divided by the magnification. Okay, so we said that was 85 millimeters divided by 425. Magnification. 425. So you just look at your drawing. Okay, and when you calculate that, you should get 0.2 millimeters. Okay, so 0.2 millimeters. And you have to include the units because you could have had it in centimeters. And B, algae photosynthesize. This process uses carbon dioxide. Hydrogen carbonate indicator can be used to determine the concentration of carbon dioxide in a solution. So the color of hydrogen carbonate indicator, if it's purple, there's a low concentration of carbon dioxide, red, there's a medium concentration, and yellow, there's a high concentration. So 1. A student wanted to use hydrogen carbonate indicator to investigate the effect of light intensity on photosynthesis in freshwater algae and describe how the student could carry out this experiment. Okay, so this is the design and an experiment question. There's always a design and experiment question, just like there's always a graph, there's always a table, there's always a drawing. Okay. So this question is out of 6 marks. You have things you can put down. You can put down the independent variable. You should put down the dependent variable. You should put down a couple controlled variables. You'll only often get marks for a couple controlled variables because there's only 6 marks in total. You list 6 controlled variables. It's not much of an experiment, is it? Something about the method. And you might want to mention something about reliability. How do you make sure it's a reliable experiment? The answer to that is always repeat at least twice. Often there's a mark for safety, This time they don't actually include a mark for safety, but often there's a mark for safety. Wear goggles or something like that. Use tongs if you're using something, a hot water bath or something like that. Okay, so let's begin. First of all, if you're investigating the effect of light intensity on photosynthesis, you probably want to change the light intensity on the algae. So change the light intensity on the algae, and that is the independent variable. Check. Okay, what's the dependent variable? That is, you're going to record the color of the indicator. And you want to say when you want to record it. So record the color of the indicator after 5 minutes. Choose an amount of time that you think would be reasonable, maybe 10 minutes. I don't know, choose a time that sounds reasonable. So that is the dependent variable. Check. So let's list some controlled variables. Okay, so something about the algae. Well, now algae, you want about the same amount of algae, or maybe you use the same mass rather than amount. Maybe you weigh out the algae. And you definitely have to use the same species of algae or the same type of algae. So you want to keep the same mass and same species of algae. You'd only get 1 mark for either of those, but those are very important. Another thing you might want to keep the same is the indicator. So you might want to keep the same volume or the same concentration of the indicator. So you can keep the same volume and concentration of the hydrogen carbonate indicator. Another thing you'd want to keep the same is the temperature of the water. So when the light's really close, you might notice that the temperature of the water gets really hot and the photosynthesis goes faster because the water is warmer compared to if the light's really far away. It's going to go slower because it's colder and not necessarily to do with the light. So you're trying to only change one thing, not the temperature of the water. Okay, other things you can keep the same. You want to make sure you record for the same amount of time or until the same end color is reached, depending on the method that you've chosen up on this step. Okay, and you want to make sure that the initial color of the indicator is the same as well. Okay, so do they all start with the same, maybe they all start with a purple color and you want to wait until they get to a medium color or a high color. Okay, it doesn't really help if one starts at a medium color, at the red color. Okay, as I said, you'll only get a couple marks for these because there's a lot of other things you need to include as well. You need to describe how you're going to do the experiment. So that's a few good marks. Let's go on to, that's the control variables done. Let's go on to the method. So the first thing you want to discuss is how to get the different light intensities. So there's a few different ways. You can move the source of the light fixed distances away. You can use different bulbs. So you might want to use different bulbs of different light intensities, or you could use a light meter just as well as one of those two methods to make sure that they are at specific intensities. Okay, I'm going to say move the source of the light. So I'm saying move the source of the light 20 centimeters away for each trial. I just randomly chose the number 20 because it seems feasible. If you're doing 5 different sets, if you're doing 3 sets, maybe you want to do 25 centimeters or 30 centimeters, but just choose a number so that you know that it's okay. So you're just saying a method. It doesn't really, you're not doing the experiment, so it's okay. So another thing you might want to say is, how are you going to actually keep the plants at the same temperature? Okay, there's a few things you can do with this. You could say use a light shield, use a screen, use low energy bulbs, or use LED bulbs, because low energy bulbs and LED bulbs tend not to heat the water up as much. What we've often done in our labs is we put a beaker of water in front of the light to absorb the heat from the light. Okay, and then if you do that with every single experiment, it'll be the same. And that method is the the heat shield. So use a beaker of water in front of the light as a heat shield. Another thing you might want to mention in the method is giving the plants time to equilibrate to the new light intensity. Okay, so let plants sit for 2 minutes to equilibrate to the new light intensity. Again, a randomly chosen number, but as long as it's the same for every single trial, it's okay. All right, and this, to equilibrate, whenever you're talking about things like enzymes or plants or something like that, often they do want you to make sure to include making sure it gets a chance to equilibrate. It comes up quite a lot. Okay, so that is pretty much the method. And now here's the free mark. I call it the free mark because it's something important for pretty much any experiment. How do you make your experiment reliable? Well, you repeat the experiment at least twice. So you repeat the experiment at least twice, right? Because they repeat the experiment twice. Because you do the experiment once and then you repeat it twice, that means you've done it 3 times. Okay, so that is reliability. Okay, that is pretty much, that's definitely 6 marks there. I would include a couple more than the 6 marks just in case something is, they only say count 2 marks from the controlled variables or 2 marks from the method, because sometimes I do, sometimes I don't. But the best way to do this question is bullet points. Figure out the sorts of things that you want to make sure you include. Try and get at least 1 from each of these sections, and then record them in bullet points. Another thing you can include, this time they don't include it, but often you get a mark for mentioning the control. So the control is the trial, the experiment where you expect a negative result, where you expect nothing to happen. Because if something does happen, then that's not really what you want. So in this instance, if you did an experiment, if you did one of your trials with just water in it and no algae, then there should be no color change. If there's a color change, well, then there's something wrong with your water or your indicator, or you have to go back to the drawing board. Okay, so the control is something else you might often want to put in there. And 2, state the name of a pH indicator other than hydrogen carbonate, which could be used to detect a change in pH. Well, pretty much any indicator will work. Well, the 2 most common ones are universal indicator or litmus. And there we go, universal indicator. And that answers that question. All right, and that is the end of this exam. I hope you have found this useful. If you did, please press the like button. And if you haven't subscribed already, we'd really appreciate it. And if you have any questions, comments, or anything else you'd like to say to us, we really like seeing your comments in the discussion section below. And have a great day.